I chose to research gene therapy helping to get rid of drug addiction in humans. What is gene therapy? Gene therapy is an experimental technique that uses genes to treat or prevent disease. Gene therapy to help get rid of drug addiction in humans would mean they could change certain genes in a human who is addicted to drugs or alcohol to get rid of the addiction. However, these therapies are still in research stages due to the fact that addiction genes are very complex. Unlike many diseases, there are many genes for an addiction. How does gene editing or therapy work? Scientists have to manipulate or alter DNA within the addiction genes. There are many different ways of gene editing or therapy, such as replacing a mutated gene that causes disease with a healthy copy of the gene, knocking out a mutated gene that is functioning improperly, and introducing a new gene into the body to help fight a disease. How does the procedure work? First, you have to isolate the gene. Next, you have to work to begin changing the gene outside of the subject's body. Once the gene is improved, put altered gene back in subject's body. Example of this experiment being done. The U.S. Department of Energy's Brookhaven National Laboratory led a study where they used genetic therapy to increase dopamine receptors within subjects' brains. This experiment showed favorable results to cocaine. Cocaine was reduced by 75%. Gene therapy will help alcoholics by taking out a certain gene that turns off an enzyme which metabolizes alcohol, meaning the person will experience very uncomfortable effects when they drink alcohol. Gene editing will be an extremely helpful and important technique to develop, especially because the medications for alcohol use, such as disulfiram, naltrexone, and acamprosate, are not widely prescribed due to limited efficacy, causing dangerous side effects, patient compliance is poor, etc. This procedure will help cocaine addicts by altering a gene that will produce variant of naturally occurring enzyme called BCHE. This enzyme splits cocaine into two harmless compounds. This will prevent euphoria, reduce motivation for subsequent cocaine use, and protect the patient from cocaine's toxic and addictive effects. It also has potential to help methamphetamine users. The researchers at the University of Arkansas for Medical Science conducted gene therapy experiment on mice. The anti-meth antibody-based therapies isolate and block the meth from reaching areas in the brain where the drug works to create a high. In my personal opinion, I fully support gene editing for addiction. Whether it completely takes away the addiction gene or it just makes you not enjoy using the drug, I think that we should do whatever we can to slow down our addiction problem in the U.S. In 2017, 20 million Americans who needed substance abuse treatment weren't able to receive it. I believe that this could definitely cut that down, especially because it sounds much more appealing than being trapped in a house with random people for months and months. It would be a quicker process and a lot less torturous. Gene therapy for addiction will be a very useful procedure because genetic factors account for about 40 to 60% of a person's vulnerability to drug addiction and 79% for alcohol addiction. Overall, the major focus is turning off the pleasurable or rewarding effects of a drug so that a person doesn't desire to keep using them. This might not create a full abstinence of drug use, but it could be useful in harm reduction in social drinking, drug use, etc.